All right. Thank you so much for having me this evening. It's wonderful to celebrate women and education. That's really close to my heart. So I'm very happy to hear like-minded women. And I'm very happy to talk to you this evening about coronaviruses. So Christine asked me to speak to you about the coronavirus and about vaccines. And I'm going to talk a little bit about antiviral drugs just because that's what I do. And I'm going to try to do it in a reasonable amount of time with a reasonable amount of depth. So if there's something that you just want some clarity on, please feel free at the end to ask me questions and I can go back and clarify. So let's just start with the picture on the slide right now. This is SARS-CoV-2. This is the virus that's causing all the trouble right now. This is an electron microscope picture. So I'm sure you might have seen pictures of the coronaviruses before. They have these spike proteins, these large proteins that stick out from the surface. So each one of these is a separate virus. And SARS-CoV-2 is, of course, the virus that's causing COVID-19, which is the what's making so many people sick around the globe. So what is a virus? Let's start there. Viruses are really, really small. I can't overstate that. They're a small particle made up of protein and nucleic acids. Nucleic acids are either DNA or RNA. And sometimes they have some fats in there as well. And that whole, so they have a protein and fat shell, and inside they have some sort of nucleic acid, either RNA or DNA. And they're so small that you can't see them under a normal microscope. You can only see them under an electron microscope. Actually, until 1930, we didn't even know what a virus looked like. We have been looking at bacteria since the 1600s, but we didn't even know what a virus was actually a virus until the 1930s. So a virus is this small entity that enters our cells. It needs our cells to make more copies of themselves. And so it enters our cells, it uses the cell to make more machinery. And just so you know, a cell is the building block of our bodies. Our body is made up of cells. And so the viruses are going to get into those cells, they're going to replicate, and then they're likely going to kill the cell when they leave, when they've made enough copies of themselves. Now, both the virus and the immune response to the virus is what could make you sick. So it's not just the virus that can make you sick, it's actually sometimes the immune response. And with this virus in particular, it's the immune response that's making people sick when the virus gets into the lungs and starts causing a strong immune response in your lungs. And that's what's causing you to be sick. So what does this virus look like? So I said viruses are really, really small. And SARS-CoV-2 is 120 nanometers in diameter. Now, 1 nanometer is a billionth of a meter. And a human hair is 75,000 nanometers in diameter. So a human hair is 75,000 nanometers. And a virus, this virus is 120. So now you can see it's so small we can't even see them with a normal microscope. The virus is actually a pretty big virus with RNA for a genome. So inside it's got the protein shell and it has RNA on the inside, and this RNA virus actually codes for almost 28 different proteins, which is really a lot for an RNA virus. Some RNA viruses make 4 proteins. This one makes 28. The reason why I'm telling you that is because that amount of proteins that virus makes means that it can do a lot of things that some viruses can't do. It can manipulate the immune system, our immune systems. It can shut things down. It can turn things on. So it has a lot more control of what's happening in your body than some other kinds of RNA viruses. So I've showed you what SARS-CoV-2 looks like, but what is it? So it's a beta coronavirus. Now, you've all heard of coronaviruses by now, and I think it's interesting that we all know coronaviruses because Coronaviruses are actually not very well studied viruses. If you think about viruses, you'll think of HIV or influenza. Those are very well studied viruses. Coronaviruses are not. Some virology textbooks don't even have coronaviruses in the textbooks. So we don't have as much information about coronaviruses as we do about some other kinds of viruses. But we know SARS-CoV-2 is a beta coronavirus. We know it's about 88 to 90% similar to two bat coronaviruses. So that kind of gives us an idea of where this virus came from, that it came from a bat. It's about 80% similar to the SARS virus that emerged. That's the language we use for when a virus shows up in the human population, so starts infecting humans. So there was a SARS-CoV-1, we just called it SARS until now, that emerged in 2002. So the SARS-CoV-2 that's circulating right now is about 80% similar to the one that came out in 2002. Now that one was interesting because you'll notice it only infected 8,000 individuals and it had quite a few deaths, 808 deaths. So it had a high mortality rate, but it didn't infect a lot of people. And the reason for that is that SARS infected your lower respiratory tract, so lower in your lungs. The SARS-CoV-2 that's around now infects your upper respiratory tract. So it's much easier to spread a virus if it's in your nose, because when you sneeze, it comes out a lot easier. So it was similar to the virus now is similar to SARS-CoV-1, but not exactly the same. And then there's another beta coronavirus that causes serious disease in humans. It's called MERS. You might have heard of it. And it came from Saudi Arabia. And so the virus that's around now, SARS-CoV-2, is only 50% similar. And now there's a word that we use for animals that can support a virus inside of them, but they don't get sick. And that's the word reservoir. And so we think that for SARS-CoV-2, the main reservoir was the bat. So that was the animal that kept this virus, and the virus stayed alive in the bat, but the bat didn't get sick. For something like MERS, interestingly enough, we think the reservoir was a dromedary camel. So a different animal kind of supported that virus to stay alive. So we think, we think that SARS-CoV-2 came from bat or bat guano, bat poop, with or without an intermediate host. And I'm going to show you again in another slide what that means. So what does this virus do when it gets inside you? So here's our virus here at the top. You can recognize it because of the spike proteins that are sticking out. You can see it as the RNA on the inside. And you'll hear in the news a lot about spike proteins. And the spike proteins are important because, for one, they're on the surface of the virus. So if you're developing a drug, you can target the surface of the virus easily. The second reason why the spike is important is because it's what binds to the cell. It's what helps the virus get in. And remember, viruses need to get into your cells in order to make more of themselves. So this virus binds to the ACE2 receptor on the surface of your airway cells. So when you breathe in the virus, its spike proteins are going to bind to the surface of your airway cells. And that's how it's like a lock and key. When it binds to the ACE2 receptor, it's like the door opens and the virus is allowed into the cell. And once the virus gets in, then it starts making more copies of itself. It copies its genome, its RNA, and it makes copies of its proteins. And then it assembles into the virus particles, and then those particles get released. So that is the replication cycles, what we call it, of the SARS-CoV-2 virus. Now, SARS-CoV-2 is better at binding to the ACE2 receptor than SARS-CoV-1, which is one of the reasons why it's more transmissible, or you can catch it easier, because it can get into cells better, it can replicate better, and it can make more of itself then. So I said that this virus came from a bat. How does that work? You might hear in the news something called if virus jumps. A virus jumps to a new, it jumps into humans. Well, obviously, you can see pictures of these viruses. They don't have legs. They can't really jump. So what does that mean? It means we use a word called zoonosis. That's when a virus mutates in such a way so that it can infect a new host. So host is in this case, an animal that supports the virus to be able to infect. And if the reservoir is a bat, how did it all of a sudden infect a human? Well, it mutated. And viruses mutate all the time. That's what they want to do. They want to mutate to, maybe when they mutate, they'll gain a new function. They'll gain some new skill that they didn't have before. So they definitely try to make mistakes when they replicate in order to mutate. And what might have happened is that in December of 2019, there was a coronavirus in a bat, and that coronavirus mutated when it was copying itself. And then the new mutated virus now has gotten a new capability. Its S protein now can bind and enter human cells. Now, at the same time that this mutation was happening in a bat, there had to be a human who got in close contact with the bat and inhaled maybe some bat droppings. And then that new virus got into the lungs of the human. And now, because this virus has this new capability, it can infect the human and start replicating. So zoonosis is the word we use for that ability to change to a new host. And you might have heard that there might have been a bat source, a pangolin, which is an awesome animal. If you haven't looked them up before, I highly suggest looking at them. They're amazing animals. There's also a little bit of news about a snake. I think we've refuted the role of snakes in zoonosis, but this is a brand new article that just came out this month. And it just shows you how complex the possibilities are for, where did this virus come from? But it is likely that there was a virus that came from a bat, the coronavirus. It may have had, so the reservoir was a bat. There may have been a pangolin involved as an intermediate host. There may have also been another mammal as an intermediate host. And then somehow we got this coronavirus that now could infect humans. So it's a complicated story. We don't have the answers, but it's likely a bat, either directly or through some sort of intermediate host. Okay, but of course, that's not the only variant or not the only mutation that's happened with this virus. Viruses can mutate a lot. It's very normal. Influenza, HIV, those viruses mutate with incredible frequency. HIV is the most variation, the virus with the most variation on the planet that we know of in humans. It has incredible mutation capabilities. Coronaviruses actually don't have those same capabilities because they have so many proteins that they code for. Their genome is quite large. They actually have an enzyme. It's called a proofreading enzyme that fixes mistakes when the genome is copied, so that you don't get a lot of mutations. So we'd actually be probably in a lot more trouble if this virus didn't have this proofreading enzyme. So, but viruses mutate all the time. And many of these mutations are going to result in daughter viruses. So you have the original virus, and then you have the daughter viruses that are produced when the virus replicates. Those daughter viruses may have no advantage, or they may be worse at infecting, replicating, or transmitting in their host. But some of the mutations that happen are going to give the daughter viruses an even better advantage. And so that is why viruses mutate, for the chance of there being an advantageous mutation. And so I don't know if you know, but the first variant of the SARS-CoV-2 virus actually emerged last January and February. So we're hearing a lot about variants right now, but in fact, the first variant came in January, February of 2020. It was called D614G. And it actually completely replaced the original form of the virus by June 2020 across the globe. So the variant that's circulating now is actually something new, different from the Wuhan variant. And the D614G is actually had more infectivity and transmission than the original strain of the virus. So this is very normal. It happens with viruses. They're going to mutate randomly, but it's actually a sort of adaptation to the new host. So the virus is just going to kind of get used to being in a human, and it's going to make mutations along the way. Now, there are some variants that have made the news. Obviously, cluster 5 emerged in Denmark in mink farms in September of 2020. And this was more of a problem for minks than it was for humans. So that one is not so much of a worry for us. But there have been 3 variants that I'm sure you've noticed that have been in the news. B117, which emerged in the UK, B1351, which emerged in South Africa, and P1, which emerged in Brazil. Now, these are important for 2 reasons. One reason is because these mutations are all in the S protein, so then that spike protein, the part of the virus that helps get into the cell. So if you get mutations in the S protein, it could be that the virus can't infect the cell at all, or it could be that the virus can infect the cell better. And so for these 3 variants, it looks like they might actually be better at infecting, or at least for the B117 and the P1 strains. So they look like they're even better at infecting humans. With B1351, that doesn't seem to be the case, but the problem with that B1351, I'm trying to avoid using geographic locations, because when you talk about viruses, you don't want to talk about where they came from, because it gives negative connotations to the places where it arose. And really, you have very little control about where a virus arises from. So for the B1351 strain, it may not have changed transmissibility, but it is affecting how well the vaccine works with that strain of the virus. So mutations not only can affect how viruses infect cells, but they can also affect how well vaccines work. Because if the new variant is very different from the vaccine, then you're not going to get protection. So, okay, so now we have these viruses. This virus, has infected humans. It has gained the capability of infected humans. It's adapting to humans. How does it transmit? How do you catch this virus? Well, it's a respiratory virus. Okay, so it infects your airways, and particularly in what's makes SARS-CoV-2 so easier to catch is that, like I mentioned, it's in the upper respiratory tract. So it infects your upper airways, which means that you're going to get lots of virus in the top part of your airway. It's much easier than to sneeze or cough and transmit it to somebody else. So that's why you could catch SARS-CoV-2 so much easier than SARS-CoV-1, for example. The other thing that's another characteristic of SARS-CoV-2 transmission is that you have 5 to 6 days between when you get infected and when you have symptoms. Usually it's 5 to 6 days. It can be up to 14 days. That window of 5 to 6 days is a problem, because that's the time where you are infected, but you don't know. And unfortunately, you are infectious. You can give it to somebody else in that window. So even though you don't know you're sick, you could be getting other people sick. Now, on top of that is the problem that there are some people who get infected and actually never know they're sick. And those are called asymptomatic carriers. So these are people who are sick, can give it to somebody else, but don't have any symptoms. And they think up to 44% of all transmission could be due to asymptomatic carriers. We believe that transmission is primarily close contact, so you have to be close to somebody, and the virus is being transmitted by respiratory droplets. So that means the virus is in fluid that comes out of your nose and mouth, and it suspends in the air, but the droplets are quite not large, not gross large, but they're bigger than an aerosol. So Aerosols are tiny little droplets that are in the air that can be suspended for long periods of time. And it's not quite clear yet whether this virus transmits by aerosols, but it definitely transmits by respiratory droplets. Another thing that I don't have on this slide, but I think is important, is that what's unique about this virus is that 80% of new cases come from 20% of existing cases. What I mean by that is most people who get this virus don't transmit it to anybody else. It's just a few people who transmit it to a lot of new people. And so you've heard of super spreaders, and that is a unique characteristic of this virus. And we don't really understand how that works, but it is something really different about this virus. Now, I just want to take a little side note here and say that masks do work. So if you're thinking, okay, this is air in the air, how do I protect myself? Masks work. And it's kind of funny because when I was first interviewed in the news about masks, when they first started to come up, cloth masks, and I said on the news, don't bother with cloth masks. They're not going to do anything. But in fact, they do something. They do help trap those larger respiratory droplets. They trap them in the mask so that they don't transmit easily to someone else. They actually also may act as a barrier the other way around to protect the wearer from air particles in the air. Now, the most effective cloth masks have multiple layers of tightly woven fabric. This is a balance. I have some masks that I'm wearing and I can barely breathe through them. They're so tightly woven. So, you have to find a balance there, but it has to be something that, you can't see through, with multiple layers. And fit is important. Please cover your nose and mouth with the mask, not just your mouth, because it's not effective if it doesn't cover your nose as well, because your nose is in your airways. And I wanted to show you this one study because I thought this was really fascinating. This was a study that looked at different states in the United States, and they compared COVID-19 cases and deaths in states that required masks and in states that did not require masks. And what they found was there are 10 times more cases in the states that didn't require masks than those that did. So the flip side of that was, in the states that wore masks, there was 90% less cases. Additionally, deaths were reduced by 80% in the states that required mask wearing. So masks work, even if they're uncomfortable and they fog up your glasses. Okay, so now we have this virus. It has adapted to get into your airways. I've showed you how it replicates. I've told you how it transmits. What if it gets inside you? What happens? How are you going to feel ill? And I'm sure everybody's aware that SARS is the severe acute respiratory syndrome. It affects your airways primarily, or at least first. So your clinical manifestations are going to be fever and cough. And then there's labored breathing, muscle pain, headache, and then interestingly, diarrhea, which is a secondary site of infection for this virus. So it can start in your airways and it can move to your gastrointestinal tract. Now, what kind of treatments do we have? Unfortunately, not very many. So if you do get this virus, you're going to be given respiratory support if you need it. So put on a ventilator. And one of the best treatments we have at this point is an antibody transfusion. So essentially, patients who have gotten better from this virus will donate their blood. The antibodies will be taken from that blood and given to other patients who are really sick, and the antibodies will help fight the virus. So there's really not a whole lot of treatments that are available for the virus. This is the Johns Hopkins University COVID-19 dashboard. This has been following the pandemic since the first days of it emerging from Wuhan. And so you can log on here anytime, it's free, and you can see what the status is globally, you know, what country's winning or losing for the infections. And you can see as of a few days ago that we're at almost 108. I'm sure by now we are 108 million cases and 2.3 million deaths. So, you know, this is a serious pandemic that we're dealing with right now. How do we protect ourselves from this on a population level? How do we protect our population? So we protect it by herd immunity. Herd immunity is when a certain proportion of the population has immunity. Immunity means they won't get sick from the virus. And when you have, so let's look at two herds, two different populations. In this herd, a lot of individuals here are healthy, that's the light blue, but they don't have immunity. There's a few individuals who are immune to this virus. So that means they've either been vaccinated or they've had the virus, their immune response has responded and they have memory. And so when people come into a herd or a population like this, where many people are not protected or not immune, you can see how quickly the virus can spread, because it's very easy for these contagious people to interact with healthy people without immunity. Now, if you have herd immunity, then you can see how many people here are immune to this virus. They're not going to get sick if they interact with the virus. There are a few people who are not immune, and they're going to be protected by all of the people who are already immune. So when this person who's infected comes into this population, it's not a high chance that they're going to run into someone who's actually doesn't have immunity. So this herd immunity protects those in the population who can't get infected, who can't get vaccinated. And so this is the basis for vaccinations, is to not only to protect the individual, but also to protect the population, as long as enough people get vaccinated. So for SARS-CoV-2, we think we need at least 70% of the population to be vaccinated in order to get herd immunity, to protect those who can't be vaccinated. Now, the other strategy of herd immunity is just letting the virus go, you know, just letting it infect the population. And Sweden tried that. And that did not work very well for them, because this virus is not a virus we know very well. It can cause devastating effects in different populations, such as the aged population. And so a vaccine is the much safer approach to get to that 70% protection in order to protect the whole, our population as a whole. So what is a vaccine? How do we get this protection? So a vaccine is either, when you're thinking about viruses anyways, a vaccine is either a live virus that's been weakened. It's a live virus. Sorry, so live virus it's an inactivated virus or an attenuated virus, something that's been weakened, or pieces of a virus, either the protein or that RNA or DNA. And it's used to stimulate the immune system. So what the vaccine is doing is it's trying to tell your immune system that this is a virus you need to pay attention to. This is a virus that you need to create memory for. So when a vaccine is first put into your arm or up your nose, or however the vaccine is administered, your innate immune response is going to detect it first. And your innate immune response is going to decide whether to respond to the vaccine or not, and how to respond. Now, once we've passed the innate immune response, it's going to activate the adaptive immune response. And the adaptive immune response includes T cells and B cells. You might be familiar with those. T cells are used to kill virus-infected cells, and B cells make antibodies to kill the virus. And so you need both of these to be activated to get a really good immune response. Now, it's the adaptive immune response that has memory, and that's what we really want to harness with the vaccine, is memory. You want your immune system to remember this virus. so that the time you actually see the virus for real, you're protected. So what is immune memory? So after your first, if you think about something like chickenpox, when I was a kid, I was taken down the street and introduced to the girl down the street with chickenpox so that I would get chickenpox, so that I would never get chickenpox again. That's memory. After your first exposure to a virus, your T cells and B cells, your innate immune system gets activated, then your T cells and B cells get activated, then most of them die at the end of the infection. But 5 to 10% of your T cells and B cells stay alive. And these are the cells that remember that virus. So when you're reinfected with that same virus, those memory T cells and B cells will activate faster, stronger, and better so that you wouldn't even know you're sick. The B cells and T cells will activate and get rid of that virus immediately. So your vaccine that you got was that first exposure to the virus. It was a safe exposure, much safer than getting the real virus. And it's going to establish memory so that the next time when you're going to the grocery store or you're talking to a friend and you get sneezed on and that virus gets into your airways, you have memory B cells and T cells that activate right away and get rid of the virus. So that's why vaccines work. Now, an effective vaccine is one that establishes long-term memory, so memory that lasts. And unfortunately, not all vaccines provide long-term memory. For example, influenza vaccines, they don't provide long-term memory. They only last for about 9 months, which is why you need a new vaccine every year. But if you get a great vaccine, you can have lifelong protection against that virus. So the real key here is that when you get infected with an actual virus, your memory B cells and T cells activate quickly to protect you so that you don't even get sick. Now, I wanted to just spend a moment here to talk to you about the difference between a vaccine and an antiviral drug. So a vaccine is something that you give a healthy person, so they're not infected with the virus, to help establish immune memory to protect you from future possible exposures to that virus. So you can get an influenza vaccine or this coronavirus vaccine. You're healthy, but your immune system is going to respond and is going to remember that virus. So if you actually see the real virus, you're protected. Now, an antiviral drug doesn't establish memory. It doesn't help your immune system remember that virus for another exposure. An antiviral drug is something that you either give before an infection, so prophylactically, or after you're infected, that's a therapeutic drug, to stop the virus. The drug may target the virus itself, or it may activate the immune response. Antiviral drugs do not induce memory, so it's not like a vaccine. Some examples of antiviral drugs you may have seen are Tamiflu is a very common one, acyclovir, ribavirin. Unfortunately, there are not a lot of antiviral drugs available because the virus, when it infects our cells, it takes on the cell as its own. So trying to create a drug that kills the virus but not the host cell is actually really difficult. So there's not a lot of drugs available. So vaccines are definitely our forefront, strongest strategy to control the current pandemic. What's in a vaccine? Well, it depends. It depends on the vaccine. This is a really great infographic from the British Society of Immunology. Most what's in a vaccine is water. After that, you're going to get the active ingredients, so pieces of the virus. And I'm going to talk to you a little bit about the coronavirus vaccines that are being available now, what's being developed, and what kind of active ingredients are being developed. But there's going to be pieces of the virus, and that's what your immune system is going to recognize. Now, that's not the only thing inside a vaccine. You also need something called an adjuvant. Now, some vaccines are self-adjuvanting, which means the active ingredient is the adjuvant as well. But remember I talked about that innate immune response that decides whether to pay attention to the vaccine? The adjuvant is what makes the innate immune system pay attention to this vaccine. If you didn't have that adjuvant, your vaccine would do nothing. It would just break down in your body and you wouldn't get an immune response. So you need the adjuvant to wake up the immune system and pay attention. There's also some preservatives and stabilizers. This is to keep vaccines safe. You see a lot of these more so in multiple dose vaccines as opposed to single dose vaccines. But there are some preservatives and stabilizers, and there also might be residual traces of things that were needed in the manufacturing. But all of those are safe below concerning levels. So that's what's in a vaccine. Do vaccines work? And I'm sure you can guess as a virologist, I definitely believe that vaccines work and that they're appropriate to control virus infections. These are just a few timelines you can see here of different kinds of pathogens. So diphtheria, pertussis, and measles. Measles is a particularly difficult virus that can infect people so easily. You know, I told you that coronaviruses, the herd immunity is about, you need about 70%. For measles, you need like 95% of the population to be vaccinated in order to get herd immunity because of how transmissible that virus is. And you can see as long as everyone gets vaccinated, that virus disappears from the population. So vaccines work. The gold standard story, of course, is smallpox. Smallpox killed 500 million people in the last 100 years of active infection. It killed more people than both world wars. It was devastating. But it's eradicated. That means it's not replicating or transmitting between people on our planet anymore because of vaccines. And polio is another one. Polio is almost completely gone from our planet because of the polio vaccine. So vaccines, of course, do work. For the current virus, SARS-CoV-2, ever since the virus first emerged and started to look like it was going to be a big problem, there were immediate responses to develop vaccines. And there's been a lot of different strategies to create a vaccine. This is a really great paper from Nature that just looks at all the different kinds of strategies. I'm just going to go through them really quickly just so you understand the diversity of the kinds of vaccines that are being produced. The first vaccine strategy is called a live or inactivated virus. Now, we don't use a lot of live virus vaccines, but we do use a lot of inactivated or attenuated viruses. This vaccine strategy is our most common vaccine strategy, for sure. What we do is we grow up a lot of virus, we inactivate it or weaken it in some way, and then you put that into somebody. This is a really good vaccine strategy because you get the real virus, you get a real immune response, you usually get really good memory. The problem with this vaccine strategy is that you need a lot of virus, and coronaviruses are really difficult to grow in culture. So we actually can't pursue this vaccine strategy very well for SARS-CoV-2. Another problem is that this kind of vaccine strategy actually causes a lot of immune response in the lungs. It's called eosinophilia of the lungs, and it's a really bad side effect. So there are vaccines using this platform in China and India, but the US and Canada have not approved these strategies for human use, at least not yet, which is unfortunate because this is our known strategy. This is the strategy that we use for vaccines, and we can't use it for this virus. So that's a problem. The next strategy, so using the real virus and weakening it, okay, we can't use that. The next strategy is, what if we take a virus that we know is safe and we genetically engineer it so that it puts spike proteins on its surface? Okay, so it is, it's a real virus, so you'll get a good immune response, but it's safer because it's not coronavirus. In a lot of cases, it's called an adenovirus. So it's a safe virus that has been engineered to make coronavirus spike proteins on its surface. So it's likely going to be safer. The problem with this vaccine strategy is that you can really only use this vaccine once per person. After they've mounted an immune response to this vaccine, the next time you try to use it, it's going to remember that vaccine platform. It's going to remember the adenovirus and it's going to shut it down. So this is not a vaccine that you can use a few years later on the same person. Another problem with this vaccine strategy is there's a lot of proteins in a virus that's not the spike protein. And so you get something called a dilution effect. So you don't get as strong of an immune response as you could with some of the other strategies. This strategy of using viral vectors or using something like adenovirus as a carrier has been used for gene therapy, but it's never been used before now for human use for vaccines. This is the platform that AstraZeneca is using for their vaccine. So this is the vaccine that the UK has developed. It's being used in the UK and is anytime going to be approved for use in Canada. Now, if we simplify vaccines even further, well, remember what a virus is made of. It's made of protein and nucleic acids. So what can we make a vaccine out of? Protein or nucleic acids. So the protein is either pieces of proteins. So this is a protein subunit vaccine. So you can take just the spike proteins or the M protein from the coronavirus, and you can put it into as a vaccine and see if you get an immune response against that. These protein subunits are they're easy to make. Well, easier to make. They're very safe. They don't usually induce a strong immune response. Now, Novavax is using this protein subunit vaccine platform. So this is the Novavax vaccine. This is the one that's going to be hopefully made in Montreal. So this will be our first Canadian-made vaccine. The way Novavax's works is they actually don't have the proteins kind of floating around like this. They have the proteins sort of sitting in a fat bubble, sort of like a virus-like particle, but not quite. And so it's a little bit different than what this looks like, but it's kind of the same principle using the spike protein as a vaccine. So then there's these virus-like particles. This is actually a really cool technology where you take a virus, you take out the nucleic acid, and we just use the shell as the vaccine. It works really well. You get a good immune response. It's got a good safety profile. They're incredibly difficult to make. You can't make them fast enough. So we actually have virus-like particle vaccines for hepatitis B and human papillomavirus, but we have none for coronaviruses right now. We don't know how to make them for coronaviruses. So that's unfortunate because that would have been a pretty nice platform. And finally, there are the nucleic acid vaccines. So viruses are made of proteins and DNA or RNA. And because coronaviruses are RNA viruses, and also because RNA is actually a little bit easier to get protein made from than DNA, the two nucleic acid-based vaccines that are currently available in Canada from Moderna and Pfizer, they're RNA vaccines. So in that case, we have a piece of RNA and, piece of RNA, and it is covered in a fat bubble. And that fat bubble is going to help the RNA get into the cell. It gets into the cytoplasm of the cell and it gets translated into proteins. Those viral proteins are the spike proteins, and they're what's going to stimulate the immune response. Okay, so these vaccines are relatively easy to make. I doubt Moderna and Pfizer would say they're easy to make, but relatively. The nice thing about these RNA vaccines is they can rather quickly accommodate variation. So remember we talked about some of those variants, particularly the one from South Africa, that looks like it's not going to be a very effective, especially the AstraZeneca vaccine may not be good at establishing memory against that variant. But with a vaccine like this, where it's just RNA, you can change that sequence rather quickly. and make a new sequence that matches the new variants. It's pretty safe. There's no virus here. It's just RNA. You need that fat coat, that lipid coat, to get it into the cell. And unfortunately, these complexes are not very stable, so you usually need to keep them at very cold temperatures. And that makes it difficult. Not as difficult in developed countries like Canada and the US and Europe, but much more difficult in developing countries. Again, this is a platform that's never been approved for use in humans. So we're brand new territory here. And this is the platform for Pfizer and Moderna's vaccines. So I just wanted to stress how amazing it is that we have vaccines this quickly for coronaviruses, because we've never made a vaccine for coronaviruses before. Influenza, we're good at. We're great at making influenza. Well, we're pretty good at making influenza vaccines. We've got all the infrastructure in place. We can have tons of embryonated eggs. We can make vaccines really quickly. So when H1N1 in 2009, when the swine flu emerged, we created a vaccine, but the virus emerged in April, and by December of that year, anyone who wanted a vaccine for that flu virus could get one, because we knew how to make that vaccine. We knew the platform. Unfortunately, we've never made a coronavirus vaccine before, which is why you can see we're trying all kinds of different platforms, all kinds of different strategies, because we've just never done it before. And there are some challenges with coronaviruses. Unfortunately, coronaviruses, when they infect humans, they don't usually give us lifelong immunity. So if you get infected with a human coronavirus, you usually have protection for 2 to 3 years. And then after that, the memory B cells and T cells die. You don't get memory anymore. So this is going to be a real challenge long term, is to be able to have a vaccine program that supports herd immunity over time. We still don't understand SARS-CoV-2 immune memory. It does seem like asymptomatic carriers don't have immunity. People who get very mild symptoms only have very short-lasting immunity. So this is going to have serious implications for keeping that herd immunity at 70%. None of the front-runner vaccines, none of the vaccines that are being used right now are on proven platforms. We've never used them before. They've never been proven in humans before. And we don't have proven safety data. Well, that's a little concerning. Although, I mean, I know that the FDA and Canada Health is working their best to keep these, of course, as safe as possible. But we are rushing vaccine development. Vaccine development usually takes 6 to 10 years, and we're trying to do it in a year. So it's moving quite quickly. And there's a lot of people working on vaccine development. This is from the New York Times. You can look at it yourself on a slow Friday night. The coronavirus vaccine tracker. And you can see there's a number of our vaccines, lots and lots, that are in the different phases of testing. And these are global statistics. So there's 4 vaccines that are approved for full use around the globe, and 4 that have been abandoned because they're not safe or they're not effective. And of course, these 2 numbers are going to change depending on how these studies go over here. The 2 approved vaccines in Canada are the RNA vaccines from Moderna and Pfizer. They both have incredible efficacies of 95%, which is, I mean, that's just amazing for a vaccine. You need 2 doses. You get your first dose, and then you have to wait 3 to 4 weeks, and then you get your second dose. And they need to be stored at very cold temperatures because they're RNA and they're unstable if they're not stored at cold temperatures, which isn't so bad for Canada, not great for some other places in the world. Coming down the line is the AstraZeneca vaccine. That's that adenovirus, that virus vector vaccine, where they've engineered a safe virus to make spike proteins on its surface. It's not as effective, but it's pretty good at 82% efficacy. You still need 2 doses. And the problem with 2 doses is that you need compliance. For one, you need that person to agree to come back, but also 2, you need to not run out of vaccine. right? Which is what we've done now. You can't give out vaccine and not save a second dose for those individuals. So 2 doses is proven to be problematic. The AstraZeneca vaccine can be stored at 4 degrees, which is much more reasonable. And then the Novavax, that's that protein vaccine. That's the vaccine that may be made in Montreal. It has about an 89% efficacy so far. It also needs 2 doses, and it can be stored at 4 degrees, so much more reasonable temperature. Okay, and I just wanted to finish. Oh, I'm not quite done yet. Okay, so I just wanted to finish to tell you a bit about the drug that we're working on. So we're working on a drug, we're not working on a vaccine. What we've done, I don't know if you remember, but I talked about innate immunity and adaptive. Adaptive has memory. Innate is what decides what to respond to. And what we did was we've put an innate immune stimulant on a nanoparticle. And this triggers really strongly, it strongly triggers the innate immune response. And if you can activate the innate immune response, you can have a broad spectrum protection. What that means is innate immunity is not specific to one virus. B cells and T cells are, but innate immunity isn't. So if you activate the innate immune response, you're going to protect not just against coronaviruses, but against any virus. So we envision this to be something you inhale maybe once a week or once a day to protect yourself. Your airway cells will be activated, and then the virus won't be able to infect. This is what we're envisioning anyways. So I wanted to show you some pictures here. These are human lung cells that we grow in the lab. And I may be biased, but I think they're absolutely beautiful. With those beautiful, healthy cells, you can see they're all individual cells. They've got nice membranes. So this is your lung cells healthy. These are your lung cells on coronavirus. They're not there. They died. They're all dead because the virus got into those cells, replicated, and then escaped by killing the cell. Now, these are those same cells, but with the drug. So this is human lung cells with the virus, but with the drug, and they're completely protected. So we think we've got something pretty exciting here. Now, this is actually 14 days post-infection. That means that we gave the drug, then we added the virus, and even 2 weeks after, we still saw no virus present. So we're seeing good protection in human lung cells. Okay, and my final slide here, I just wanted to reiterate what you can do to protect yourself against this virus. Remember, it's an airway virus, so cover your face with a mask, wear something that fits well to your face. That's going to hold the virus in to protect others and hopefully protect you from the virus if it's suspended in the air. Stay 2 meters apart. We know definitely from studies that the virus is much more easily transmitted when you're close to somebody. Okay, so stay at least 2 meters apart. Sanitize and wash your hands frequently, and get vaccinated when you can. And that's the end of my presentation. Thank you very much for listening.